Hi, I'm Andres at ModernVivo. In this video demo, I'm going to show you how our literature intelligence platform can extract key in vivo experimental conditions from millions of open peer-reviewed papers. I'll also show you specific features surrounding drug candidates, clinical trials, as well as regulatory documents and responses made by regulatory bodies. Stay tuned to learn more. The first thing you'll see when you log into ModernVivo is your homepage. This allows you to see your usage, how many papers you've searched, as well as your top models and recent searches. If you click on recent searches, it'll take you to the results. But before we look into the results, let me show you how to build a search. Navigate to the search builder on the left-hand side. The first step in building a search is to enter keywords. These can be keywords you want to include in your scan or keywords you'd like to exclude from your scan. So in my case, I'll reset this. I'm interested in type 2 diabetes mellitus. So I'll type in T2DM for type 2 diabetes mellitus. I'm interested in mice models and GLP-1. Enter, and these search words are included in the scan and connected with AND operators. If I want to exclude certain terms, let's say I'm not interested in rat models, I'll exclude that using the exclude button. And now that's added with an AND not operator. You can get more creative with your Boolean logic. So for instance, let's say I wanted to look at T2DM and, quote, type 2 diabetes. I can enter that like this. I'll open a parentheses, click after this first keyword, type or, hit enter, and then with quotations, type 2 diabetes, and then close. Boom, and it's added. So in real time, ModernVivo estimates how long your search will take. And you can see that happening on the right. It'll also tell you if your search is too narrow or too broad. If too narrow, likely you won't find any papers that match your search. If too broad, you'll probably need to add more keywords in order to dial it in. So for instance, if I typed in some gibberish, obviously that won't be found in any paper. It'll say, there's no papers that find that. Now, if I only typed in mice, for instance, it would tell me the search is too broad. So I'm gonna go ahead and edit this directly, remove this gibberish, and save the search criteria. So that's step one of building a search. Let's move on to your extractions. This is where you can select the different fields of information to be extracted from papers. There's always going to be a base set of fields that are extracted from every paper we look at. So these are your core assays, your core experimental factor fields, and your core intervention fields. They also include animal species, output measures, and products. In addition to these, you can select up to 10 animal background and maintenance extractions or experimental condition extractions. So I'll just choose a basic set that I think are typically relevant. I'll choose animal age, animal sex, weight, genetic background, and the study duration. In addition to that, I'll also choose the administration timing, intervention dose, as well as the route of administration. And that brings me to 8 selected fields. I think this will suffice, so I'll move on to the next step. The final step is to give your search a name. In this case, I'll call it T2DM, type 2 diabetes mellitus, GLP-1 in mice. And additionally, as you scroll down, I can also select how many papers I want to process in this search. So depending on your subscription level, you'll have more or less papers to budget. And let's say I don't want to use all 372 papers in this search. Instead, I just want to look at 30 papers. So I can use this slider to select how many papers I want to search for. Now, I'll also filter so I know which papers to prioritize in the search. So in my case, I want papers from the last 2 years, so namely between 2024 and 2026. And I want to see the papers that have been cited the most first. And those are the ones that will be returned in the results. You can also look at the papers that have been cited the least or the oldest publications versus the newest publications. It's your choice. Now, if you're going to look at all the papers, this doesn't really matter. You can filter these in various ways when you review your results. And that's it. That's your search. And you hit submit search, and this will go into a queue, and the tool will work in the background while you do whatever you need. Because we're a web app, you don't have to keep this open. You can close down your window, go into the lab, run some experiments, do whatever you need. So I've already run the search, and you can see the results down here. So I'm going to terminate this, just so we don't have to do it again. And that's it. That's how you build a search. Next, let's review the results. So the first thing that you'll see are these paper cards here on the right-hand side. Each one of these represents a unique peer-reviewed paper that was found to match the keywords and extractions that we selected for when we were building the search. In case you ever forget what those are, you can always click on this drop down to see them again. Here are these keywords for type 2 diabetes, as well as the extracted fields broken up by fields that pertain to animal background and maintenance, and fields that pertain to experimental conditions. Really handy in case you need a reference. So at the top, you'll see the title of the paper, as well as the journal in which it was published and its publication date. We've also included these handy tags to help you see whether or not the interventions cited in the study were approved or not. You can also see a tag to show whether or not there are FDA review documents. I'll talk a little bit later about the FDA regulatory documents and other regulatory documents that we give you access to. In the top right, you can also see the number of citations that the paper has. So at first glance, you can see each paper is broken down by studies. So this paper specifically had 3 studies, namely a DB mouse glucose lowering study, a mouse intestinal distribution study, and a rat oral absorption study. And for each of these, you can see here on the left the experimental groups for that study. And you can click through them. You'll see slight changes in the extractions. And for each of them, you'll also see the assays and output measures. This is really key. At a glance, this gives you all of the critical information you need to design a study or understand a study without having to do any of that heavy lifting and sifting through walls and walls of scientific text. So if I click across the top, you can see that the extractions for animal age, genetic background, and so on and so forth change, as well as the output measures. Right? So previously, we looked at a glucose tolerance test, bodily fluid glucose assay, and others. In this next study, we have fluorescence detection assays, as well as immunofluorescence confocal microscopy assay. So this is really important and really the bread and butter of this software platform. For each paper, you can also dig a little bit deeper into the paper details. So clicking this purple button here allows you to see the same extracted information, but scrolling down will help you see the methods summarized by the tool. So you can at a glance really understand what was the approach that scientists took in this study to find their results. And then the results are also summarized for you. But you can also go back to the original papers as well. So if you click View Paper, or if you go back to the previous screen and click on Original Paper, this will take you to a new tab where you can go to the original publisher's website. So while this search only shows 30 papers, sometimes your searches will be pretty large, actually hundreds of papers, and it can be really difficult to parse through that information. And so we've created a number of handy filters for you here. So on the left-hand side, you can see these. You can filter by model. So in our case, there were 32 different models cited across all of these 30 papers. You can also filter by the drugs mentioned in these papers. In our case, there were 2 main drugs. You can also look at these papers based on the extractions, right? Not always will every extraction be mentioned in each paper, but if you're specifically curious about, let's say, animal sex, you can look for papers that only show, let's say, male mice. So really handy if you really want to narrow in on a specific extraction field and just find papers that look at that field. We can also look at affiliations of authors. So whether or not the paper came from academia, government, and industry may be meaningful to you. You can also filter by publication dates. Perhaps you want to look for precedent that is more recent versus older, or vice versa. And you can also see unavailable or excluded papers. So these are typically paywalled papers that we are not able to parse out using our tool, but we will show you those papers in case you want to pursue them otherwise. And finally, you can see your favorite papers for those that you had starred, as well as papers that were cited in downstream clinical trial publications. In this case, there were none. Some other handy ways to filter the results. We have a really interesting decision tree feature. What this allows you to do is walk through from disease, animal species, to induction factor type, the induction factor, assay, and finally the output measure, in order to create a chain of logic that you can use then to filter the papers, right? So let's start this from the top. Let's say we want to look at alcohol withdrawal as kind of your disease indication. It'll show you what animal species were cited in all the animal alcohol withdrawal papers. You click on those, and then it'll populate the induction factor type column with the different induction factor types. So we click on exposure factor, and then we have 2 options for the induction factor. And then finally, the assay, and then the output measure or measures. And so by selecting these, you can systematically filter papers without having to do all of the manual toggling of that filter page that I showed you before. And you can just click View Papers, it'll clear your previous filters and just show you the papers that match this line of reasoning. So a really powerful tool to help you think through how these studies were designed and built up. We also have the ability to look at the different models cited across all the papers. We can also look at drugs. And so another way to filter is by looking at the different drugs that were included. So given this set of filters, you can see that there were 2 drugs here, and you can view papers, clinical trials, and regulatory docs for those. So let's dig into those actually. These are really interesting features that help with translation. So first, the clinical trials tab. So you can see over 5,000 clinical trials here. The columns indicate which drug they refer to, the title of that clinical trial, the indication, the phase, the NCT ID, as well as links to clinicaltrials.gov, where you can learn more about those studies. Really handy if you want to see how these studies performed. In addition to that, we also have a really powerful feature called regulatory documents. So this allows you to see all sorts of different documents, from pharmacology reviews to toxicology reviews, approval documents, and ultimately you can see the data that was submitted to regulators and how they responded. So we've included really handy links to all of those, so you can access that data through the FDA, and it'll take you to a separate PDF where you can learn more. And last but not least, we have a summaries feature. This is to help you pick out trends over time among the literature. So in this case, I generated a summary for a specific type 2 diabetes mellitus model. You can generate summaries for any model here. You can see for each extraction, they show you the most common choices that were made, a breakdown of those choices as a percentage of the total papers, and trends across time. So for instance, let's say you saw an experimental condition that was highly cited between years X and Y, but then between years Y and Z, the field shifted. You can see that here in these trends. So really handy to get a high-level overview of how the literature has progressed or changed in meaningful ways. And last but not least, we made it really easy to share all of this data with your team. So if you click Export and Share, you can create a shareable link that allows anybody, regardless of whether or not they have a ModernVivo account, to view the data and interact with it just like we did here. You can also export the data if you want to do your own analysis outside of the platform by clicking Export in CSV. This concludes the results. If you have any questions, please follow up with us. You can send us an email at hello@modernvivo.com and we'd be happy to chat. Thanks for listening.